So today I'm going to present you a research I did with a small paper I made. It's about antibiotic resistance data and how we can use SOSA ontology to store this data. So it's in the ABRomics project, which is a project financed by the French National Research Agency. So what is antibiotic resistance? So antibiotic resistance is a major health hazard for human, environment, and animals. The cause is that the resistance genes, or the genes that are responsible for antibiotic resistance, are very mobile, and so they can be transferred to a species to another, and to an environment to another. So we needed to have a national platform that could track the propagation of such antibiotic resistance genes. This is why in France we developed the ABRomics platform, which is the French antibiotic resistance surveillance platform. So basically, the objective here is to provide a platform that allows researchers and clinicians to upload their sequencing data. We also provide workflows that analyze these sequences, and relevant information such as the resistance genes are then stored in the platform. So the goal here is to create an interoperable database containing the annotated multi-omics antibiotic resistance data to make it fair as possible, and to be allow users to perform different workflows on their data. So basically, currently, the platform is composed of a website where users upload their data, and then workflows can be triggered on this data, and then at the end, the workflows produce other data that are stored in a relational database. So everything is stored in a relational database for now. So this, of course, presents different limits. As you know, the analysis data are not interoperable with other data sources, which may make very difficult to share our data with other teams. And it's also difficult to benefit from other published knowledge graphs, such as UniProt, for instance. Also, one major issue also is that when we deal with bacteria, we usually use graph algorithms for bacterial communities. So having a knowledge graph would which will allow us to use this kind of algorithms. So we built a knowledge graph, and I will explain you how it works. So first of all, we checked for different ontologies because we didn't want to create any terms. We didn't want to create any other ontology. We wanted to reuse everything and to glue the things that we found interesting. So we have ontologies that are relevant for represent omics data, to tag omics data. So the arrow ontology, which is the antibiotic resistance ontology, and Go, you probably know of it. The NCBI taxon for taxonomy, and also to represent clinical and environmental data, we used NCIT and ENVO. However, these ontologies are very good when it comes to tagging data. However, it's difficult to represent the data that are generated by the workflows. So to counter this issue, we use the SOSA ontology, which stands for Sensor Observation Sample and Actuator, which is an ontology that is used in sensor networks. So another domain completely. However, it was very suited for our use case because it provides 4 concepts that are very interesting to us. First of all is the sensor. So the sensor can be considered to be the instrument of measure. So it would be the sequencer. The observation is the result of the observation by the workflow. The observable property would be the sample that is observed, so it would be the sequence. The feature of interest would be the specific characteristic we want to observe. So it could be a resistance gene, the length of the gene, the coverage, etc. So to validate the instantiation we made of this graph of this knowledge graph, we asked biologists to different which kind of competency questions we can use to measure this performance. So we have the first one, which is what are the most represented antibiotic resistance gene in a specific region of interest. And so we have the notion of of resistance genes and of in space. And the second one was what are the actually actively circulating antibiotic resistance genes given a specific time frame for at least 2 different sample types. So here in the competency equations, you can see we have the notion of multimodal data and time and space. So here you have the schema for to represent the metadata of a sample. So we used for the provenance, you can see we have the provenance with time, space, the sensor, and the researcher. And we use also domain ontologies to represent the taxon and the tissue on which the sample has been collected. So we use, in terms of ontology, we use prov a lot here. And as you can see, we have a notion of the time and you have the notion of time and the provenance. And then here you have the interesting stuff. We reuse the SOSA ontology to represent multimodal data. So for instance, we have the sample 1, which would be, for instance, the sample collected on a blood sample, on which we perform, we have different multiple observations, observation 1 and 2. For the first observation, it was generated by the workflow 1. The thing that was observed was the resistance gene. And as a result here, the result of the observation is the name of the gene. And we also have a timestamp. And of course, the sample has been observed multiple times by the same workflow. And the same workflow also made another observation, which was focused on what is the strain present in the sample. And here we have the numeric value, which is the abundance of this strain in the sample 1. Afterwards, to validate this, to validate the instantiation, we tried to answer the first competency questions. So have put here the Sparkle query. So first of all, we get all the genes. Afterwards, we select only the antibiotic resistance gene observation. Then we only select the gene nodes. And afterwards, we use the Wikidata to only filter the genes that were, the samples that were collected in a specific country, here in France. And so making this query a federated query. So we also made a small demo. So you can, if you have a link here, so you can check the work we made. For now, we only have the two competency questions on the demonstration, but in the future, we will implement more. So it's still a work in progress. So we want also to add support for GeoSparkle in order to support the use cases such as finding, for instance, selecting only the samples along a river, for instance. So having when we don't have clusters of samples, like for instance in city, we have samples only in specific spots. We want also to support environmental data, so the data is collected on a large area. So GeoSparkle would be very useful for this. And we also want to support environmental and metagenomics data by annotating this data with ontologies such as ENVO and NCIT, and also supports more One Health scenarios. Yeah. So thank you for your attention. So if you have any questions, let me know.
Thank you. Why did you introduce ABRomics :ABRI gene as an EURN instead of an IRI? It seems a very strange technical choice. Is that by accident in the typo, or?
Where?
The second green line, yes.
Yes.
That means, because you have a square, the angled brackets around it,
yeah.
Means it's not a prefix, but a scheme, EURN scheme. So it means it's not a HTTP IRI, but a completely new way of fetching data that has not been registered anywhere. So I was wondering, is that just this angle brackets, is that a typo, like you forgot to remove them, or did you have a technical decision there?
No, I don't remember when I put I think I do the reference to...
I hear typo in the backup.
Yeah.
Okay.
Maybe Alban can...
It's great that it's a French initiative, and I really love anything that has to do with antimicrobial resistance. Are you also connected in a European network, or is it just French?
It's just French, I guess. Yeah.
You have comments, I see. Maybe this is more of a discussion then, or?
No, just to comment, it's a project that has been funded by the French government. So it's a national platform, but we are participating into the Elixir network of bioinformatics facilities, and we are participating in all the microbiome communities. So we are aiming at participating to collective works in the context of Elixir first. Yeah.
And it's not an or, I would say, continue the discussion. I have one final question, if there are not any others. I was curious, I saw you using NCIT, you're using various ontologies. And one specific ones, when you want to model France, you use Wikidata, while France is also covered in NCIT. Is that
it's in NCIT?
an omission, or is it, was there a design choice for using Wikidata over NCIT?
No, I didn't thought it was in NCIT actually. Yeah, it is just because I knew that in Wikidata we have all the countries, so I wanted a data source for all the countries with no exception. So maybe in NCIT there is not all the
Jervin and I shouldn't ask his questions.
Actually,
just
I don't
look
know.
at the details.